And now for the second part of our discussion on epithelial tissue, we're going to move into discussions on not necessarily what epithelial tissue looks like and how to identify it, but different compartments of the cell. When we look at a three-dimensional structure, we're going to have, and you can think of it as top, bottom, and the sides of these different three-dimensional compartments. So with a cell, it's not going to be any different. So by the end of this unit, we're going to identify those compartments. And specifically, we're going to be looking at the different structures that you'll find on the apical surface, the top and the bottom versus the sides. And we'll define the molecular components of each one of these specialized structures as it relates to adhesion, as it relates to diffusion, as it relates to absorption and anchorage of cells. So in this illustration, we can see the top of the cell. You can see the rounded dome-shaped surface. This, of course, as an epithelium, is either going to be the covering or the lining epithelium. And this is the apical or the free surface. From this aspect of the cell, if we move along down the side, and here you can see the side of two adjacent cells, this is referred to as the lateral portion. Now, we often consider this to be continuous with the bottom of the cell, which is the basal compartment. And so you see the term basolateral described there because we have structural complexes that are going to functionally and structurally separate molecules and receptors on the apical surface from those on the basal and the lateral surface. Because of the fluid mosaic model, these receptors and proteins can float between basal and lateral. But with these structural adhesions that we see at this point, we'll be describing these in much detail. Receptors on the lateral surface cannot diffuse into the apical surface and vice versa. So they are distinct from one another. So the two functional compartments are the apical and the basolateral. So on the apical surface, we're going to see some specializations, and one specifically that we'll be talking about are called microvilli. Now, villi are finger-like projections, and micro just refers to the fact that these are small in nature relative to the size of the cell. And as we look at these structures, they're only going to be localized on the apical cell surface, and they're going to be present for transport and primarily for absorption. So I think you can already in your mind realize that in our intestinal tract, our alimentary canal, this is where we're going to find a huge percentage of cells that are going to have microvilli on their apical surfaces. Now, that absorptive function is going to be enhanced by the fact that these microvilli increase the surface area. And as we look in our alimentary canal, the microvilli, as well as other specializations of the organs themselves, are going to assist in increasing the surface area up to 600-fold. Now, as we increase surface area, as you remember from physiology, that's going to increase the rate of diffusion, and that's how absorption happens in our intestines. Here we have two differing views of microvilli. On the left, we have a scanning electron micrographic view, and then on the right, we have a transmission electron micrographic view. These are going to be finger-like projections, and that's typically how they're described. They're also going to look possibly like the bristles of a brush. So often these microvilli are referred to, especially from the light microscopic level, as resembling a brush border or brushed border. Now, these finger-like projections of microvilli are going to be comprised of molecular components, and internally they're going to be 20 or 30 microfilaments. Now, these microfilaments are going to be filamentous actin, and we know from our discussion of muscle from anatomy and physiology, F-actin is comprised of the globular actin or G-actin components. These are going to be running longitudinally parallel with the direction of the microvilli. Now, you can think of these as supportive cables that are standing up, supporting the length of that microvilli, and they're going to be cross-linked and stabilized by other proteins, such as fibrin and vilin. Now, these microfilaments that are cross-linked are secured inside this finger-like projection of the plasma membrane with binding molecules such as myosin 1 And calmodulin, we've seen calmodulin and its effect and function along with calcium, but it's also going to function here in the stabilization of the microvilli. Here we have an illustration showing villi at the terminal end, or the apical end of the microvilli. Here is the cell toward the bottom. We have our filamentous actin. Then you can see our fibrin, cross-linkers, and other molecules. Now, this is a very simplified view, understand. Then we're going to have actin running along perpendicular to the actin of the microvilli. This is going to be the terminal web that we have. We also have intermediate filaments, part of the cytoskeleton. And this is how these cytoskeletal components anchor themselves to the cytoplasm of the cell, then extend into the microvilli. Now, this terminal web is going to anchor those microfilaments via myosin 2 and spectrin. Again, these are cross-linking elements that are going to tie in the actin of the microvilli to the actin of the terminal web in the cytoplasm of the cell. Here we see a transmission electron micrograph. Actually a scanning electron micrograph. This is the microvilli. We can see the cross-linking of our terminal web of actin. This is the plasma membrane that we see along the surface. Some of these smaller fibers are spectrin, but most of these long cable-like structures are going to be our terminal web of actin, and you can see how we have our microfilaments extending down from the microvilli, perpendicular microfilaments running through, creating the terminal web and anchoring them in place. Now, our second apical specialization, this I've always felt like was something of a misnomer, and it's why we put this second. You may be thinking already of cilia, and we will be talking about those. That's going to be our third apical specialization. But we're going to start with this structure called stereocilia. Now, these are extremely long. These are going to be much longer than microvilli, which tend to be the shortest in height of our apical modifications. But stereocilia are not cilia at all. Make sure you make a note of that, and I need to highlight this and put it in bold. Stereocilia are extremely long microvilli, and they're only present in the body within regions of the epididymis of the male reproductive tract and the sensory hairs in the cochlea that are going to help us sense balance and movement, basically acceleration and deceleration. So stereocilia have the same internal structure as microvilli. The distinction is these are much, much longer in nature than you see with microvilli. Here we can see a cross-section through, actually longitudinal section through the epididymis. Microvilli may extend up about that far from the cell surface. Maybe the thickness of the arrow itself would be that brush border. Here you can see the microvilli have extended much further into the lumen of the epididymis. And here we can see how thick and elongated these stereocilia are in this scanning electron micrograph. This is all going to be stereocilia and material from the testicles as well. You're going to have spermatozoa in here as well. This is an example of some stereocilia that we find in the cochlea. Almost an artistic look to the cells and then the stereocilia that we have present in the cochlea. They're arranged in rows, and how these stereocilia, how they bend and push against one another, are actually going to send signals to the brain about acceleration and deceleration, part of our equilibrium system that we have in the body. Now, our third apical modification, cilia, these are going to be distinct from the microvilli. Microvilli are present mostly for absorption. Our stereocilia are going to be present for some absorption, but also for the sensory detection in the cochlea. Cilia, however, are present for motility. These are present in our respiratory tract. They're going to help remove debris, and they're going to be able to bend and wave to move material throughout the body. We'll see these in respiratory tract. We have cilia present in oviduct and other areas. On the left, we can see a scanning electron micrograph that's been pseudocolored. And on the right, we have a longitudinal section through cilia in a transmission electron micrograph. And we'll be looking at the internal structures of these. They're quite distinct from what we see with our microvilli. So the internal structure of the cilia, if we're looking at a compare-contrast, that's one of my favorite types of questions. If we look at the internal structure, microvilli are comprised of microfilaments. Cilia, however, are comprised of units of microtubules. And as we organize the microtubules, they're going to be present as a structure that's referred to as the axoneme. We're going to have two central units surrounded by 9 doublets. Now, this is the appearance we're going to have as we look at a cross-section through a particular stereocilia. Now, these 9 doublets, or peripheral doublets, are going to be composed of an alpha and a beta subunit. The alpha subunit is essentially going to be a ring of 13 protofilaments, whereas the beta subunit is going to be an incomplete ring, or a C shape, that's going to be adjacent to the alpha subunit. They're going to be closely opposed to one another. We'll see this in the next slide. Each one of these peripheral doublets is going to be secured back to the central doublet by a radial spoke. These proteins are going to look like the spokes of a bicycle wheel, and that's how we got that name. And that's going to help to synchronize the movement and the sliding action we're going to have of all of the microtubules, of all the subunits at the same time, to coordinate the movement of the entire cilium. Here's our illustration. We can see in longitudinal section our peripheral doublets and how they circle around our central doublet pair. And of these elongated microtubules, when we cut them in cross section and look at that cut surface, this is the structure that we see, and this is the axoneme. And this is the axoneme. Here we see our central doublet, and then our 9 peripheral doublets composed of the alpha and the beta subunit. Here we have our radial spokes, and we're going to talk about these other cross-linking agents as they're tying this entire structure together to help the cilium bend. Now, as we look at the base of the cilium, we are going to lose our central doublet, and we are going to be left with our 9 triplets that surround that empty space. This is referred to as the basal body and has the same organization and structure as our centrioles that we learned about during mitosis and meiosis. This is a microtubule organizing center, is going to help to organize the microtubules and help to form the axoneme in the more distal portions of our microvilli. Once again, here we see another illustration. We can see almost in a relief 3-dimensional view our 9 peripheral doublets, central doublet, alpha, beta. There we have our radial spoke, and then how that is aligned in our cilium. Basal body, often referred to as the kinetosome. Basal body is still accepted in almost all literature. And then we can see how by the movement of some of these molecules relative to one another, that can bend. Now, the real motor that drives the bending and the movement of the microtubules is going to be a microtubule associated protein, or a MAP, that is called dynein. Dynein is going to anchor adjacent microtubules, as we can see in this illustration. And dynein also is going to be able to bind ATP, cleave ATP, and release that energy so that it can change its conformation. And based upon the shape of dynein at rest, when you release the energy, you have, now this illustration is referring to a power stroke. Do not confuse this with the power stroke in muscle contraction. But we change the conformation of dynein. Based on that change, we slide one microtubular filament over the other. And as you do this and coordinate it with our radial spokes and our other anchoring filaments, such as nexin, which is going to be more of an elastic type protein, you can see how we stretch it, and once we release this conformation back to its original shape, this elastic material is going to rebound those actin microtubules back to the original location. So as we saw earlier in our illustration, we have our axoneme throughout the bulk of our microtubule. However, at the base, we're going to have a nucleolar organizing center, a microtubule organizing center, such as a centriole, and that is going to be our basal body. The 9 triplets surrounding an empty center. We don't have that central doublet pair. We simply still have the radial spokes in the organizing proteins, and that's going to be at the base of a particular cell. This is typical transmission electron micrograph view of cross sections through cilium. Many of these are going to be slightly hazy, out of focus, but I think in this one you can see our 9 peripheral doublets, our central doublets, and even have a slight inkling of radial spokes within this image. There you can see, often we will use these highlighted images that go back and forth, flipping one to another. You can see the outline of the doublets, the central pair. We can see the alpha and the beta subunits on many of these, the radial spokes. So these are often useful, and we'll see these in labs as well, to try to help us realize where these structures are. And as we flip back and forth, we can see the labels, then click off, see the actual image. Hopefully you'll find these extremely helpful. I call these the red line maps for a lot of our histology. Now for the lateral portion of the cell, what we're going to see is we have junctional complexes that are primarily going to function to adhere adjacent epithelial cells to one another. They're also potentially going to form what amounts to a zip-lock seal that's going to prevent any material in the apical or luminal compartment from diffusing or spilling down between the cells and into the underlying basement membrane. There is no place this is more important than in our gastric epithelial cells, where we have hydrochloric acid. The apical surface has protective mucus that nullifies and buffers that hydrochloric acid, but that's not present in our lateral and basolateral surfaces. So the hydrochloric acid would destroy and basically digest those epithelial cells. So these junctional and functional complexes are referred to collectively as terminal bars. These are light microscopic views that histologists saw very early on. And these terminal bars are comprised of these more apical junctional complexes. They certainly are on the lateral surface, but you see they're much closer to the apical surface than they are to the basal surface. So these terminal bars that we see present are going to be comprised of these junctional complexes, the zonula adherens, the zonula occludens, and then we're going to see these that are scattered throughout, the macula adherens, and then other junctional complexes that are present for communication, and these are the gap junctions. You should be fairly familiar with these from our discussions of cardiac muscle and to a lesser degree with smooth muscle. But we're going to step through each one of these lateral specifications to look at their structure and the function of these junctional complexes in the lateral compartment between epithelial cells. So the zonula occludens, zonula means belt, and occludens means to shut up or shut tightly. So these are referred to colloquially as tight junctions. When you look at our terminal bars, they're going to be the junctional complex that's the most apical of all of our junctional complex structures. Now, as we look at adhering two adjacent cells to one another, remember our plasma membrane is comprised of phospholipid bilayers. Well, imagine that the outermost layer of those phospholipids, in this case, are going to interdigitate one to another. So instead of seeing 4 phospholipid layers of cells pushed up against each other, you're only going to see 3. You're going to see the 2 innermost layers that face the cytoplasm of each cell, and then essentially a unified outer, and this one's going to be in the very middle now, that outer layer of the phospholipid that's fused to the outer layer of the adjacent cells. So these are fused or welded together. That's going to be the tight aspect of this. Nothing is going to leak past that apical compartment, past that tight junction, because there is absolutely no space whatsoever for material to pass. Now, this fusion occurs because we have essentially cell adhesion molecules that are present as transmembrane receptors. Now, these are often referred to as occludins and claudins. Now, these are CAMs. They're calcium-independent cell surface proteins. They're going to have a cytoplasmic component that anchors into the cytoskeleton, a transmembrane hydrophobic region, and then the extracellular region that's going to be able to form adjacent adhesive complexes with the occludins and the claudins of the adjacent cell. Now, to add to this junctional complex, we have calcium-dependent adhesion molecules that are called cadherins that are going to function in a very similar way. Now, as we look at this illustration, we can see that we have a cadherin. They're often abbreviated with the first letter of the tissue in which they were identified. So E cadherin is the epithelial type of cadherin. We also have an N cadherin, which basically refers to a neuronal type, but they're very similar. We can see our cytoplasmic portion of the receptor, the hydrophobic transmembrane portion, and then the extracellular portion that, along with calcium, forms a relationship with a cadherin on the adjacent cell. These are both E cadherins. And you can see in this illustration how we have anchored that cadherin into the cytoskeleton, this being the actin part of the terminal web, with this complex of molecules that are referred to as the catenins. The catenins are going to bind to the cadherin. They're going to bind to actin to anchor this protein to the cytoskeleton of the cell, and so there's no way this protein is going to pull through that phospholipid bilayer. They are attached to each other, and this distance is so short, the phospholipids, I mean, we can see it in this illustration, of course, for our educational purposes, but these are so close together that these phospholipids are going to fuse with this. So you won't actually see 4 distinct layers. You'll see one. This will be a collective one, and then we'll have a third one on the bottom. The same can be said on this side. Here we have occludins, which are illustrated in red, and they're going to bind in a calcium-independent manner to the occludins on the adjacent cell. Other molecules are going to form structural complexes to cross-link the occludins with our cytoskeleton. Now, this is a very interesting scanning electron micrographic view. What has happened is we have taken tissue and we have rapidly frozen it in liquid nitrogen and other cryoprotective agents. And they have freeze fractured. Basically, you apply a force and you are going to shear the tissue along the crystal plane of the ice. And what we see is we essentially have microvilli, these finger-like projections, and this is one of the cut surfaces, but we've sheared this in such a way that we can actually see the tight junctional complexes of our terminal web, or terminal bar. And what I want you to appreciate here is the fusion that happens between adjacent cells. It's not just one straight line. You know, it's kind of the analogy of the Ziploc bag, but you have this network of these fused elements of the phospholipid bilayer, and they're quite thick. So I think you can appreciate even one layer would probably do a fairly decent job of protecting the apical material, prevent it from flowing down to the lateral surface. But this very complex structure and all of these fused membranes are absolutely going to prevent that from ever happening. Now, in our transmission electron micrograph, we can clearly see the dark lines and the lighter material between. This is our lipid bilayer of one cell. This is an extracellular space, and then we have 1, 2 dark lines, a light line. This is the lipid bilayer of the adjacent cell. And as we follow this up, you can see that extracellular space diminishes, 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 and finally, there we have the fusion of our internal, the external layer of the phospholipid bilayer. We can see the from cell 1. Here's from cell 2, and then the fusion that occurs. There's a fused part there, a fused component there, lightly confused there. And that is going to be that part of our zonula occludens. Again, the most apical of our junctional complexes, forming a very tight junction to prevent material from spilling from the apical compartment to the lateral compartment. Here we have an illustration. These are cells that are grown in culture, and I want you to also appreciate that these structures are in fact a belt that are completely circumnavigate or go completely around the cell. So not only do they have a height and thickness apical to lateral as it's moving down, but it also completely goes around the cell. So no material is going to pass. Let's say we're in the apical compartment looking down. There is no chance for us to move from this compartment between the cells to get anywhere near the basement membrane or the lateral component. It's just not going to happen because of the zonula occludens. You've been occluded from moving into that area. Now our second component is going to be the zonula adherens, as in zonula belt. Adherens means to anchor, to hold on to one another. So here in our terminal bar, we've got our zonula occludens. Now here's the zonula adherens. Now this is going to have different structure, and it's also going to have different function because the zonula adherens is there for strength of anchorage, to hold the cells tightly adjacent to one another, not to prevent materials from moving in this case. Now the molecular structure is also going to use cadherins. And remember, cadherins are going to be calcium dependent cell adhesion molecules. We're going to see that same structure. We're still going to utilize the catenins. An additional molecule, alpha actinin, is going to help to anchor the cytoplasmic component of the cadherins to the actin cytoskeleton. And so as we look at the structure of our zonula adherens in this illustration, we can see our tight junctions. You can see the fusion of the membranes, and slightly more basal as we move downward. This is the apical surface. We can see that the membranes move apart, and here we once again have an extracellular compartment, but we're going to have fairly dense material on the cytoplasmic face, and we're going to see these cytoskeletal components moving away from this area, and this is going to be a cross-section through our zonula adherens. Once again, we've got our transmembrane cadherins, calcium dependent. We have our catenin and alpha actinin structures that are going to anchor and cross-link the cytoplasmic domain of the cadherins with the actin cytoskeletal web. So these are microfilaments that we see, actin microfilaments, in this particular illustration. Here we have a red line illustration for our zonula adherens. And as we look down the cell, here's our two adjacent cells, cell 1 and cell 2. We are going to see that as we move back and forth for these, we can highlight our zonula adherens. In the upper right, it's going to be quite an extensive area. Once again, as we look, we can see the greater density of material adjacent to the plasma membranes, but we still see this space, this cytoplasmic space. That's going to be maintained by the distance of the cadherins one to another. And that's going to be the second portion of our terminal bar. The zonula occludens most apical, the zonula adherens is going to be next on that list. Now notice in the slide we have something called the fascia adherens. The fascia adherens is going to be similar in its structure, the molecular components. However, it is not essentially going to completely encircle a cell. And cardiac muscle is our example of this. The fascia adherens is simply going to be present on portions of the cell with adjacent cells to help anchor them one to another, but it's not going to completely go around a cell. Cardiac muscle is going to have a different structure. It's not arranged necessarily in layers like we think of our epithelium, but more lamina, and we'll talk about that when we get to cardiac muscle. But that we refer to as a fascia adherens. Now, as you look just a little bit to the right, you see this structure, the macula adherens, or a name desmosome. First thing I want you to notice about this is it is much smaller in size than our zonula adherens. Notice they both have this adherens name. They're both going to function in anchorage. The zonula is a belt. The fascia is a large, expansive connection without completely going around. And the macula adherens is essentially going to be a spot. And so we think of the macula adherens or desmosomes as spot welds. These are extremely strong adhesive buttons that you're going to find scattered throughout the lateral compartment between epithelial cells. Now, once again, we're still going to use a very similar molecular design for these. We're going to have transmembrane proteins that are going to anchor to the cytoskeletal elements. They're going to form junctions with other proteins in the extracellular compartment. So that's going to tie the cytoskeleton of one cell to the cytoskeleton of an adjacent cell in a very, very tight, closely fitting spot or spot weld area. Now, the proteins that we are going to use are essentially going to be similar to what we've seen with our cadherins and our cell adhesion molecules. We refer to these as the plakophilins and the plakoglobulins. Now, when we look at these structures, these are going to be on the cytoskeletal side, and these are going to be the cross-linking agents that cross-link, much like the catenins. They anchored the cadherins to the actin cytoskeleton. These are going to form these complexes that are going to anchor our transmembrane molecules to the cytoskeleton. Now, here's a compare-contrast for you. Whereas we saw actin with the zonula adherens, we are going to anchor the receptors to keratin intermediate filaments. They're often referred to as tonofilaments. This was a light microscopic term that was used for decades and decades, but we know now it's the keratin or cytokeratin intermediate filament population inside our epithelial cells. Now, the cadherin family members are going to be referred to as desmogliin and desmocollins. So these are calcium dependent, have a very similar structure. Their primary structural sequence of amino acids is going to be similar, so that places them into the cadherin family. But the desmogliins and desmocollins are going to span through the plasma membranes, form the extracellular relationships with the other desmogliins and desmocollins on the adjacent cell, and the plakophilins and plakoglobulins are going to anchor those cytoplasmic portions into and with our keratin intermediate filaments. Here we see our illustration. We've got the desmocollins and desmogliins, our cadherins. They span through the membrane. Here we have our plakophilins and plakoglobulins, part of this little spot plaque you see in orange. And here we have our intermediate or tonofilaments that are cross-linked because of the plakophilins and plakoglobulins to the desmogliins and the desmocollins. So once again, what we're familiar with, the zonula adherens and how differently it looks to our macula adherens. Once again, we have our dark plaque. We can see some of these filaments just in the lower left in this illustration, but very, very dark plaque material spaced between, but it's much smaller in size than we see with this very expansive zonula adherens. Also, we're going to see these much lower down in our lateral compartment of the cell. I know it's hard to distinguish where exactly we are in this cell, but you will be able to do that on some of the other illustrations that we have. Just giving you as many looks as we can. Now, our last junctional complex that we have in the lateral compartment is one that functions for communication. It's going to allow the cytoplasm of one cell to be continuous with the cytoplasm of the adjacent cell. So this is not for strength. It's not to prevent movement of material from one compartment to the next, but it is to allow and facilitate the movement of materials from one cytoplasm to the other cytoplasm. And we find these extensively. We find them in epithelium. We've already mentioned cardiac and smooth muscle. To some extent in neurons. We talked about these quite a bit in human physiology. But a gap junction, or a nexus, is another name for a gap junction, is comprised of 6 proteins. And these proteins are referred to as connexins. And they're going to assemble on each side, each particular cell in the appropriate location. And you're going to form this little tunnel. It's going to have an aqueous pore. And the collection of the 6 proteins is called a connexon. Now, as you line up your connexons, so here we have a connexin, Here we have a portion of a connexon, which is going to have to be 6. So here we have our connexon from one cell. And you can see in cross-section, this would be a portion of one connexon. Here we have a portion of one connexon. And as they line up, they're going to create an aqueous tunnel from this cell through the gap junction into the adjacent cell. So these must line up for that to be facilitated. Now, what can pass through a connexon? Well, we're going to look at molecules that are in this neighborhood of size. 1.5 to 2 nanometers is the diameter of a typical connexon. And that's going to allow ions, amino acids, cyclic AMP, hormones, basically molecules smaller than 1 kilodalton. This is a globular protein size, in fact. So when you look at what can pass, we're going to be moving potassium. We're going to be moving sodium, and relative to action potentials, depolarizations, excitatory and inhibitory potentials, we know those ions can move and form part of that syncytial unit. But connexons aren't just holes. They can be regulated. So in some, at low pH or high calcium, you're going to completely close them off. And vice versa, with high pH and low calcium, you're going to open those up. So don't think of it just as an open hole, but think of it in some instances as a regulatable means of communicating between one cell to another. Now we finally get to the basal compartment. We're actually at the bottom or the end of the story. And the one structural functional unit we have is essentially half of a desmosome. It's going to have the exact same structure as a desmosome, but we don't have an adjacent cell to attach to. We're going to attach to the basement membrane. So that's what's referred to as a hemidesmosome, or half desmosome. We're still going to have the placophilins and placoglobulins. We're still going to anchor into the keratin filaments, the tonofilaments, but rather than the desmoglians and desmocollins, we're going to have integrins. These are going to be our transmembrane proteins that are going to anchor into the keratin filaments, be cross-linked by the placophilins, placoglobulins, and they're going to bind to extracellular material such as laminin and type 4 collagen. That's going to anchor the cell into that first layer of our basal lamina, the lamina lucida, and type 4 collagen in the lamina densa. So here we have our illustration. We can see our plaque. We can see our keratin intermediate filaments. Here are our integrin molecules, and integrins are dimers. You're going to have 2 of these functioning together to anchor. And in this case, we are showing a relationship with laminin in green. Here we have collagen 4 in blue. And you can see how we have essentially anchored the cytoskeleton, these extracellular proteins, with the cytoskeleton of this cell. Now, laminin, type 4 collagen, they're also going to be cross-linked with other material to our type 1 and type 3 reticular fibers. And so if you think of this as a skeleton, what we're essentially doing is anchoring the internal skeleton to our extracellular skeleton. So we are creating a very strong attachment of this cell to the extracellular environment. It is going nowhere. Here we have a transmission electron micrograph. We realize we're inside the cell because these little black spots are going to be our ribosomes. We are in an extracellular compartment. Now, this is going to be the cytoplasm of another cell out here, and we're not going to worry about that at this point. But we also know we are extracellular because we can find lamina densa, lamina lucida, and then we have our plasma membrane. And as we follow along, we can see our plasma membrane, and then we find these dark densities along that basal surface adjacent to the basement membrane. Here we have another. And within this dark plaque of tissue, we can see these filaments radiating away. So these are our keratin intermediate filaments. These are our placophilins, placoglobulins, and then we have our integrin molecules radiating through here, anchoring into those components of the basal lamina.